Hi, my name is Damian, and I'm a senior scientist at Swiss Bioinformatics Institute, and I will talk about string database. So, what is string database? It's a protein-protein interaction database and a functional annotation tool. It's a one-stop shop for interaction data and functional annotation. And sources of these interactions, so if you query one protein, you will get a network of the protein and its 10 most likely interactors. And then this is derived from computational methods like co-expression, gene neighborhood, phylogenetic profiles, gene fusions. And experimentally derived, this is from mostly IMEX consortium and from BioGrid and from Intact. And they are from curated databases, that is pathway database like KEGG, Reactome, and we also do text mining, including NLP on the whole PubMed and PubMed Central. So we have also we provide both functional association and physical association, and all prediction and benchmark in one way for all of them the same way, and that's why we know we can assign the confidence score, and it means exactly the same for each of the sources, independent if it's derived from text mining or experiment. The score means exactly the same, and all the underlying data can be examined. So here you would have on the top you would have a text mining viewer where we highlight which protein, and we also in the new version which highlight the word that the NLP was focused on when talking about this, when the sentence talks about the interaction. And then for each experiment, for each high throughput experiment, or for each interaction with the experiment, we can actually derive the confidence score for it. So if the experiment is actually random, we would know it wouldn't come up in string. So and this is for low throughput and high throughput experiments. And of course, the view is highly customizable and interactive. So, and we also, whenever user queries database, we also provide the enrichment. So this is protein-protein interaction enrichment, and then also functional enrichment in 16 different categories, including gene ontology, pathway databases, domains, diseases, phenotypes, network cluster. These are like non-supervised Go terms that we derive from the structure of the network itself. And we also have the sets derived from the text mining. So we know which paper mentions which protein, and we treat them as a Go term. So we can enrich for that. So you can find your own experiment, for example. This is how we present. This data is in the single view. And you can query that with single, multiple protein. You can query with sequences. And you can query also string with the whole experiments. If you have a value for each protein attached, then we treat your experiment as a background and we randomize inside it, and you will get to the different views. So not to the network view, but for this. And then we can we enrich it, but it's a different way of enriching. This is like we empirically derive the value based on the randomization, and here we also echoes your data set and the values distribution within that set, and we of course enrich it. And this is actually paper that the set is derived from, so it finds itself, and because they mention all the proteins that are on the top, on the bottom, and we actually highlight the proteins distribution of these proteins in that particular term or the paper in your data set. So they in the paper mentions both up and down regulated proteins as their result, and we could find it. And you can click on it again, and you can get to the network view, and we show you which proteins are upregulated, downregulated within that term. And then you can query a string with COGs, so the protein families. And the newest feature, you can query string with pathway, process, publication, phenotype of disease. So these are based on these annotations. For example, you can write the Alzheimer, and we know all the curator ontologies that mention Alzheimer. And here you have KEGG, monarch phenotypes, UniProt keyword, tissues, diseases, And also, we look at the PubMed titles, and we know which mention gene sets and which mentions in the title the disease. So the first paper is the Alzheimer's disease, for example, here, and it mentions 23 proteins, and we can click on that, and we can see the network of these proteins on the left and enrichment. And because of the synergies of all these data sources, we know, for example, where these proteins are associated. These are in medial temporal lobe. And also we know that these proteins are associated also with reading comprehension, for example. So these curation efforts like add the value to each other and to understanding of the user data sets. And without any efforts from the user, it's just automatic. We map them together, and you see how they synergizes and how they corroborate each other. Yeah, and we do that all for more than 10,000 species. So we actually re-annotate all the species that we choose to incorporate in the string. So you have all of these for all of the species. Of course, if it is plant, you wouldn't have annotation with brain. So then they don't have all the 16, not all species have all the 16 categories. And yeah, so, but yes, exactly. So, but we still get a question always, where is my species? And this could be very specific bacterium or some plant. And then we did a survey with SIB that actually shows that 20% of the users didn't find their organism string, which is staggering. And so we never would be able to keep up with the pace of how the sequencing efforts. So that's why we build a pipeline. It's a string protein annotation. Is built in within the web interface. You click it here in the main page, annotate your proteome, and we just basically add your proteome to string. The only thing you need to do is to provide name. It could be anything, and provide what is actually a clade of the species you are adding. It could be as specific as a strain, or it could be as unspecific as a order, or this is some proteome bacteria that I don't know what is it, but these are annotated. So we take this account, that this uncertainty of the clade. And then you just upload a FASTA file. And then inside, we know what is identifier, what would be gene name, what is the description. So if it's some common FASTA header format, we would be able to parse it and actually echo later. You will, if you provide a nice gene names, there will be nicely named inside the user interface. So we don't yet predict them. But yeah, so we will echo everything to you, and you just click continue, and that's it. You can annotation lasts few hours, but you can go and sleep. You can come back in a month. You can close the windows. You don't need to. We would know that this is you, and then we'll say, okay, it's done. So it usually lasts, depending on the size of the species and what species is it, how close to human it is. Its annotation could be fast or slow. So from 2 hours to 6 hours, more or less. And then while you sleep or having tea, string computes all alignment against all 60 million proteins we have. We derive orthologs and paralogs at all applicable taxonomical level. So in total, we have 1,600. And so we basically built kind of approximation of the gene tree for each of the protein you have provided. And then we predict functional and physical interaction network based on evidences from all other species that are in string, and the same functional annotations from all other species that are in string. So all of them contribute to the annotation of your species. So of course, if it's very far away, probably there will be nothing that is contributed, but in principle it can, like especially very conserved family. So let's go through one example. For example, this would be controversial but important crop species, oil palm. it was actually uploaded by one of the users, so that is our example. But also, I chose it because there is the website Palm Explorer, which gives you all the of the palm oil proteins, but it also gives you the manually derived annotations for fatty acid biosynthesis. So there is someone there on this consortium annotate the proteins that are involved in fatty acid biosynthesis because it's important. It's a oil palm, and on the right you see there are references and there is protein list. So when you upload the species, you will get to this page, and you can just select your species on the input page, that is, and then you can just copy paste this Excel file into string, and we gives you the network of that process, and we indeed discover that it is annotated all by one protein with fatty acid. So the one protein is actually not homolog of the that I know of the fatty acid biosynthesis process of any protein in Arabidopsis that would be annotated with this term. So I don't know if it's false positive or not, but we got them all. But what if, of course, you don't know the process? You can simply write fatty acid biosynthesis in string, and we got all the proteins that we discover, and there are more of them because they are very close homologs to the other proteins from that process. So, and you can do it for every process in any species, and it's and it's just one click. It doesn't require any effort. Yeah, and of course, because we have that synergy, you can see that it happens. This process happens in chloroplast or in chloroplastoma, and this is free and shareable. Everyone can go to this link and see and look at this particular genome like it would be natively in string, like we would add it. It just has a name that is strg whatever, and this is available through graphical user interface, as I described, through programmatic access, and of course you can download all the data, including the annotations for that species, for your species, and you can publish it or whatever. And then the feature is 9 months old. We don't really advertise it. It's just there on the input page. And we get almost 1,000 proteomes uploaded on the top of 10,000 that we already have a string. And every month we basically have more and more, including even if there's no vacation time. So as always, we have less user, but it's still they upload more and more, and these are from 500 distinct users, and 25% of users uploaded more than one proteome. So this is really the lowest bound how many people find this feature useful, because not only they found it useful enough that, and in addition they found another genome to actually upload. So this is not the feature that you will upload 10 or 20 genomes, just your one favorite. That was the assumption, but actually lots of people have more than a genome. Up to one user uploaded up to 100 of different mycobacterium species. So, and this is the division of what is actually uploaded. And very interesting is plants. Plants is actually quarter of it, as you see, and this is interesting because actually the plant users is a fraction of our total users. So this is it shows you like this community is really underserved, we think, especially in the annotation realm. So yeah, so acknowledgement. So specifically, I want to acknowledge from Swiss Bioinformatics Institute side, Raja, which basically took like around 20 different prediction methods and put it in a one pipeline and built this system that is relatively robust, so the user doesn't have to care about anything. It just communicates between the server. There's a computing server, there's a string server, and it's all works seamlessly and is opaque to the user. It's just, yeah. And then, of course, the string is a collaborative effort with Copenhagen, with the Lars Jensen group in Novo Nordisk, Center for Protein Research, and a Bohr group at EMBL. So thank you.